our little lesson on two-trait Punnett squares, sex-linked Punnett squares, and pedigrees. So I wanted to teach you a trick for doing the two-trait Punnett squares really quickly. And you can also use it for 3 traits, 4 traits, 15 traits. Anyways, so we have in our example here um, a pig or 2 pigs, and they tell us that black is dominant to white. So we're going to say big B is black and little b is white. It also tells us that tall is dominant over short. Oops, let's put a little t there. Okay, so these are our alleles. And it says the first pig is heterozygous black and heterozygous tall. So he's heterozygous for both traits. And then whereas our other pig is homozygous recessive for both traits. And next, we would find the parental gametes. So this would be like the parental cross over here that I'm just highlighting right now. We would find the parental gametes. Okay, and so in our sex cells, we can only have one allele for every gene. So our first parent, we could use the FOIL method. And so we could have big B, big T in one sex cell. We could have big B, little t. We could have a little b and a little b and a big T, or a little b and a little t. The other parent only has little b's and little t's, so they can only make one type of sex cell, little b, little t. So in this situation, it would end up being a Punnett square that is a 4x1. Okay, so in terms of like what it would look like, I'm going to just make a little table to kind of show you here. It would look something like this. And you would put, theoretically, if all this worked out for me, let me see if I can add a column to the left. There we go. So you would put the little b, little t allele here, and I'm going to actually see if I can delete that. Make it white. There we go. So that's kind of deleted. Oops, there we go. We'll delete that one as well. Okay, so that looks a little bit better. So we have little b, little t on top, and then we can put the other parent's gametes on top. Big, if this works out for me here. Okay, big B, big T. Okay, you can also have big B, little t, and little b, big T, and little b, little t. And then you would just fill them in. If you have a capital letter, you would put that one first. You keep the B's together and you keep the T's together. Okay. And so we get, I'm just filling it out. And then for, in terms of like what work do I need to show, we would probably ask for a phenotypic ratio. So we would list the phenotypes from most dominant to most recessive. Okay. So I'm looking for black and tall. Oops. How many of those I have? I'm also looking for black and short. So it's dominant trait, dominant and recessive trait. I'm looking for recessive dominant. So that's white and tall. And then lastly, I'm looking for our white and short, which is recessive. Okay. And so if I go back in here, this box would be black and tall because it has one big B and one big T. So I have one of those. Oops, one. This box would represent a pig that's black and short. So that's one of those. This box would be a white pig that's tall. And this one would be, oops, I messed up my Punnett square here. Would be a white pig that's short. Okay, so I have my ratio is a 1 to 1 to 1 to 1 ratio. Okay, so ratio would be 1 to 1 to 1 to 1. Okay, oops. Here's the shortcut. Okay, So you could take the probabilities of coloration and height individually and multiply them together and get the same phenotypic ratio. So we can look for the coloration ratio. Okay, so it's big B, little b, crossed with little b. What are their chances of getting these same phenotypes? Okay, so I'm going to kind of prove to you that we get the same thing. Okay. Okay, so I'm going to delete my numbers here we'll go back over it and show you that you do get the same answer. Oops, let me just, there we go. Looks a little bit better. Okay, so here's my shortcut, like I said, and I think your book calls it the multiplication rule. Okay, so you can find what are the chances if you did this Punnett square of getting black coloration, and so it would be 3 out of 4 to be black. Okay, and then to be a white piggy, it would be 1 out of 4 chance. Okay, so if you did the Punnett square. On the other hand, if you did the height Punnett square, it was heterozygous tall crossed with short, and you would end up with tall being also 3 out of 4, I believe. Was it 3 out of 4? Oh, sorry. I got my stuff wrong. It's 2 out of 4 and 2 out of 4, which reduces down, of course, to 1/2. Okay, sorry. See, this is why I should draw my Punnett squares out. Okay, that's better. And then we have tall. There's half a chance that the offspring would be tall. So that's equal to 1/2. And then short, also 1/2. Okay, and if you need to draw out the Punnett square, there's no harm in that. Like, as you can see, I just made the mistake myself. So anyways, we're now looking for being a pig that's both black and tall. So we're just going to multiply our ratios together. Okay, so there's 1/2 chance times 1/2 chance, and that's going to equal 1 out of 4. Okay, and when we do this for the rest of them, you end up getting 1/2 chance of being black again. And then we saw that it's 1/2 chance of also being short. Okay. So you get 1/2, which would equal 1 out of 4. Okay. And then moving along, same thing. Okay. 1/2 times 1/2, which equals 1 out of 4. And 1/2 times 1/2, which equals 1 out of 4. So as you can see, the ratio is the same. It's 1 to 1 to 1 to 1, or there's a 1 in 4 chance, which is the same thing we saw here. Out of 4 options, there's 1 out of 4 that would be black and tall or black and short, etc. Okay, so this is the shortcut, and you can do this with 3 traits. Okay, so I would like you to try that on your own. Okay, what if we had, we'll use some of Mendel's traits. Red is, big R is red, and it's dominant over white. Okay, and we'll use little r for white petals, okay, for a flower. We can also use plant height. Big T is for tall plants, and little t is for short plants. And we can talk about the flower position, if it's on the end of the stem versus like off to the side of the stem. So he said that side flowers are called axial, and it's actually dominant, whereas the flower located at the end of the stem is called terminal, and it's recessive. Okay, so if you had a plant that was big R, heterozygous for coloration, okay, and height, but had terminal flowers, and you crossed it with a plant that was exactly the same, okay, I want you to try and see what you get as a ratio for having a offspring that's red, tall, with terminal flowers. Okay, so I'll give you a minute to do that. Not really, you could just pause the video. And you should end up with, and I did this ahead of time. I did all my work here. Okay. And you should get 9 out of 16 chance. Okay. Hopefully you can see my work. Okay. Moving on, I wanted to talk a little bit about, so basically the answer is 9 out of 16 chance. Okay, wanted to talk a little bit about sex-linked traits. So sex-linked traits are carried on either the X or Y chromosome. The Y chromosome is so small that it does have fewer traits overall than the X chromosome, and the X chromosome is essential for life. You cannot be born without an X chromosome. So you need to have at least one, okay? And there are a few traits linked to the Y chromosome. For example, some men grow hair on the outside of their ears. That's called hairy pinna. That's Y linked, okay? But typically they like to focus on X linked traits. Some of the more common X linked traits are hemophilia, which is a blood disorder, a blood clotting disorder that was really common in the royal family because of all the inbreeding, as well as colorblindness is sex linked. And we watched a video last year about a little boy named Lorenzo Odone. Some of you probably watched it as well. And he had adrenoleukodystrophy, which was sex linked. So in sex-linked traits, they're more prevalent in males than in females. These traits are recessive, and so you would have to acquire two recessive traits if you were female to show the recessive disorder. However, in males, because males have only one X and only one Y, if you have one defective gene, you have the disorder. Okay, so we're going to do a quick hemophilia problem. Okay, and so you have to include the sex chromosomes when doing these problems, or you will get the problem incorrect. And it's not just me being picky, you actually will not get the probability of having a daughter with a disease or a son with a disease correct if you leave off the X and Y chromosomes. Okay, so this is question 2 from your packet. Okay, so we'll do this one together. If a boy's father has hemophilia, so we know that the father, because he's male, he has to be XY. Okay, so that's his genotype for being male, and he has hemophilia. So maybe I'll step back 1 second and let's define some alleles. So you can be X big H, and it should be a superscript H. Okay, maybe I can do that quickly. Let's see. I can. Okay, so it should look something like that. Okay, and maybe I'll zoom in. Whoa, that looks a lot nicer. Nice and zoomed in. So that is the genotype. So X big H is the allele of the normally produced gene that doesn't cause hemophilia. It means that you're able to produce the clotting factors. Okay, so I don't like, let's put this, normal functioning gene. Okay, whereas X little h, okay, is the hemophilia gene. Okay, again, I'll try and go change this. Because this is located on the X chromosome, you would put the letters only on the X. It is not located on the Y, so Y is just Y. Okay. So anyways, Dad has hemophilia, so he's going to have this X little h. Okay, that's dad. And then mom says that she has one gene for hemophilia. So she's considered a carrier. Okay, so she's going to be X big H and X little h. Okay, so that's mom's genotype. And they want to know what are the chances that the son will inherit the disease. So they already know they're having a little boy. Okay, so that's one of the things. So you would draw your Punnett square. And let's see if I can draw a table for us. Sorry, it's taking me a little longer than I had hoped. So we would put dad's genes or dad's alleles in one box. Okay, and he has a Y. We would put mom's alleles up top. Okay, and then we're just going to fill in the boxes. So, oops, I should really put this one first. Okay, so the first child could be a daughter. Okay, so she's X big H, X little h. She would be a carrier, but she would not have hemophilia. Okay, the next scenario would be a daughter. Okay, so also X little h, but sorry, she would have hemophilia. She would be X little h, X little h. Okay, because she got a defective copy from each of her parents. In the box below, we would have X big H, Y. Okay, so this would be a son. Okay, and then in this box, we would have X little h, Y. Okay. And so as you can see, the boy has inherited this disorder from his mom, but mom is not colorblind. And dad is colorblind. He's able to pass it on to his daughters. One's a carrier because the mom's X big H masks it. And so if this was the daughter that was born, would not have the disease. However, this daughter would be colorblind. Okay. But they were asking about sons. Okay. And so just looking at the boys here, what are the chances that the boy will inherit the disease? It's a 50% chance if they know they're having a son. If the question asks, what's the chance of them having a daughter with normal vision? Okay. So then it's okay. Well, we look at the daughters. What's the chance of the daughter having normal vision? Well, 50-50. Okay. And then they can ask, what are the chances that their child will have hemophilia? Well, these two would have hemophilia. So it's a 2 out of 4 or 1/2 chance that they would have a child. So just be careful how they phrase the question. Okay. Moving on to pedigrees. Okay. And so this is part 9, and I would like to look at question number 2. Again, I'm just kind of doing all twos today. So a few things. This square, okay, is for a male. Okay, so males are represented by squares. Females are represented by circles. This horizontal line connecting the male with the female is a relationship line. It used to be called a marriage line. This vertical line is showing that they had children. They had 4 children, and we read them from left to right. That's their birth order. And so they had 2 boys first. The boys are older, and the girls are younger. Okay. I can also see that they shaded in the square or the dad. That means that he has the disorder, okay, or the trait. And then we can half shade in carriers, okay, and sometimes they don't do it for you because they want you to think about it and figure it out. And then here we can see that we need to figure out the genotypes the best we can. And so it's a little bit of like problem solving. Okay, so for dad, I know that he has at least one dominant gene, and I'm going to use the letter A. Okay, and I'm actually going to, um, let's see, I think I drew it out for us. Okay, so I'm going to kind of just like write it down real quick. So I know that he has at least one big A. Okay. Okay. I know mom because she's not shaded in. Um, and this is a dominant disorder, as the problem said, that she has to have two little a's, because even if she had one copy, because this is autosomal dominant, one copy or two copies, she's going to be fully shaded in. There are no carriers in this scenario. Okay, so this is what we have so far. Okay, all I know for sure is dad has at least one big A and mom has two little a's. Looking at their kids, I know that the sons, what their genotype is, I know that they both have to have a little a. So I'm going to write that down super quick. Okay. So this tells me that they had to get one little a from mom, okay, but they had to get a little a from dad as well. So that it's a little bit of detective work I did. I can figure out that dad also has to have a little a. Okay. um, with the same line of thinking here, I know that mom has to give an allele to each daughter. Dad has to give an allele to each daughter. So because they're fully shaded in, I know that they have to have at least one big A from dad. Okay, so I'm going to draw that in or write that in. Okay. And mom only has little a's to give. So I know that both girls are heterozygous for this trait. But even though they're heterozygous, they're fully shaded in, they're not carriers. Okay. If they were a carrier, we would half shade them in. Okay. And they would look like this. If it was a recessive disease, we would half shade like this. Okay. If someone has passed away in a pedigree, unfortunately, we cross them out. Okay. Something like this. They're just like crossed out. And then if they, maybe there was a relationship that didn't work out, there was a divorce maybe, we would put a slash through this relationship line right here. And so it would look like this. Okay, so that's showing a divorce. Okay, so I'm going to stop our video here. I hope it was helpful. Please do problems part 6, 7, and part of 9. Do the pedigrees. I know there are a lot of them, but they hopefully go pretty quickly. We will do the gene linkage on Monday. Okay, thank you. Bye.